my seminar for today. Okay. So I want to remind you that after seminar we will have lunch with the speaker in the Joyce Room. So if you want to join us for that, that would be good. Also want to remind you that at 7 o'clock tonight in this same room will be our third lecture in memory of World War I. And our own Reverend Dr. Father Watson will be delivering a poetic literary address as to what happens when poets become soldiers or soldiers become poets. I forget which way that works. Okay, so you want to come out tonight for that. That should be a good discussion of things. Who came out to the last World War I? See, was that good? I don't want to comment on that. All right. Okay. Also, next week, we will get back together here. So that's the 30th. Next week, October 30th. We will have for our guest a geotechnical and pavement engineer. So all those geology students and civil engineers and people of that sort will want to come out for that. That'll be Eddie O from the Department of Engineering from Texas A&M and Commerce. He'll be delivering our talk next week. But today we have a very special talk on the color of life and the molecules of life, given to us by Francis de Sousa, who's down here from UNT. And Dr. de Sousa is an interesting guy, very accomplished. He has over 300 some publications to his credit. So that's a big wow if you're an academic. Okay? So he certainly knows how to get things published. I guess it helps that he sits as editor in a number of journal magazines, so he can get his own stuff published that way. But I guess you got to publish something before you can become an editor too. So that kind of works out well. He grew up in India, which got his bachelor's degree and his master's degree from universities in India, and his PhD from the Indian Institute of Science. He came over to the U.S. here to do a postdoc. He did one at the University of Houston and one someplace else in France. Dijon. Dijon. So he has multiple global experiences, as we call it, on this campus. So you'll appreciate that as well. And the talk that he's giving today is a talk that he's given as an ACS lecturer, and those talks are always very good, and I think that you will enjoy very much. And so again, I would encourage you, if you have any interest in the sciences, or India, or France, or in Houston, or things like that, you want to join the speaker at lunch. We will be in the Joyce Room. Remember that room that you always wondered who was in there, right? That's the room we'll be in. So you can come and explore that, check out the napkins and things like that. We don't get the fancy cloth napkins like the president does, but we still get nice napkins, right? So you want to check it out. And that'll be after the seminar today. So let's first then welcome our speaker.
Well, I take this opportunity to thank Gary to invite me to come over here. So, when I agreed to come here, I thought it's maybe 20-30 minutes drive. Then I realized, it's a lot of driving. But anyhow, so this is a lovely campus, and I'm glad all of you are here. And today's talk, like Gary said, it is not very technical. So this says something about the molecules that make color. So that means it's very general. And if you have any questions, you can talk to me after the seminar, and I'll be very happy to answer those things. So my talk begins with asking a question. So I'm showing some pictures. Okay. The question is, what do these things have in common? Okay. So we have fuel cell vehicles. Okay. So which runs on fuel, which is mainly hydrogen. And we have something called photodynamic therapy. Okay, so there you use light and some molecules cure cancer. And we have, all of us have this, red blood cells. And we also have organic conducting polymers. Okay, so this is organic wires. And then we have green plants and algae. And then even we have a lot of chemical catalysts. Okay, catalyst to suppress the activation energy so that you can make a lot of chemical products, in a cheaper way. Okay, and then we have organic photovoltaic cells to harvest solar energy into electricity. And finally, even we have, vampires and werewolves. So what do these things have in common? Okay, so if you do not know the answer, so they all have a simple compound which is called porphyrin. Okay, so the story of this beautiful compound started a while ago. Okay, so here is, your Texas blue bonnet, something like that. This is an indigo plant, and this has that purple flower. So the assignment was to extract pigment out of this and, have purple clothes and things like that. So it becomes so expensive, okay, because it is extracted from flowers. So only very important people can, use that. Okay, so that was not very affordable to common people. Okay, so then the task was assigned to chemists, because chemists basically are nature lovers. So they don't want to kill the plants, extract the flowers and things like that. They want to make things in the lab. Okay, so in the late 1980s, okay, long before your grandparents were born, okay, there were a couple of German companies, okay, their assignment was to make a chemical which would give that indigo color or the purple color. Okay, so while doing so, they always had a side product. Okay, and that side product had this purple color. And that compound was very stable, even if you heat it at 500 degrees Celsius. Okay, so it was stable. Okay, so they were all curious, what is this purple compound which is so brightly colored and it is so stable? So finally they solved the structure. Okay, and it came out to be this molecule. So they called this porphyrin. Okay, so this is in the beginning of 19th century. Okay, so this is one of the side products. But still, chemists, like I said, they want to master the synthesis. Okay, and try to come up with a better synthesis method so that they can make these molecules in bulk. Okay, and seek out their applications. Right? So with that, somewhere in 1950s, it took about 50 years to master this art. Okay, so these are two American chemists, Rutherford and Menotti. So they did this simple reaction. Okay, the reaction is so simple. You take pyrrole, this is called pyrrole, and you have aldehyde. Okay, you condense them. Okay, cook them. Condense them means you just mix it in some solvent, heat it, and then you have this beautiful molecule. Okay, so this is about 50 years ago, and since then there are lots of papers, hundreds and thousands of papers published on studies based on this molecule. Now often, this is what the reaction I was just telling. So reaction is very simple. Pyrrole and aldehyde, you separate the products, purify them, and then you have this beautiful molecule. Now, oftentimes people say, chemistry as such is a couple of 100 years old. Okay, so there are a lot of new advances. Every day you see there are new things are developed. So often people do question, hey, what is there to research? It's already 100 years old molecule. Okay, so aren't they all pretty much the same? So this is the question often people ask. So the answer is actually no. There are many things one can do even with this base compound. Okay, so there are a lot of structural modifications we can do, such as, see, for example, this is the porphyrin we have, and inside there is a hole here. Okay, when you have a hole, that means chemists are very creative people. Okay, so they can fill this hole by using different metals. every time there was a metal, it had different properties. Okay, every time. And then organic chemists, they don't like metals. Inorganic chemists like metals. Organic guys want only organic groups. So we can do organic chemistry on this molecule so that we have different substituents on the end, and these substituents could be at different positions. Okay, and so happens, we can put metal and substituents also. So all these different substituents and metals made this research field so interesting, and people wanted to see whether there is any biological significance of this molecule. Okay, and then also, people started doing chemistry, very innovative chemistry. Okay, instead of base porphyrin model, they started playing with the positioning of these carbon groups. So instead of one carbon connecting the pyrrole rings, they had one directly connected with two carbon bonds. So these are all structural isomers. That means their carbon count is same, okay, but the way it is arranged, they are different. So every time you have such a thing, you have a different molecule. And also, if you do not like nitrogen, so we can replace nitrogen with other elements, like, sulfur, for example, or selenium, things like that. And if you do not like this molecule, you can expand them. Okay? You can expand them. So we can do so many things. That's what that field grew up in the last 100 years. So there are thousands of papers every year people publish. Okay? So that makes this field very interesting. Number one, why this porphyrin chemistry is so interesting? Number one, they are really big. Okay? Size of Texas, for example. Okay? They can contain different metal ions. They are very stable. Importantly, they can take an amazing variety of roles in the real world, like the first few slides I have shown. Okay, so which is why it has been highly researched and used. Okay, so what are the uses? Like I was telling, so it is a metal binder. It's in solar cell for light harvesting, and it is electron transfer medium, like conducting polymers. Okay, sensors and catalysts, and drugs in photodynamic therapy. Okay, so there are a lot of applications of these things. So what I'm going to do is show where in nature this type of compounds are found and their utility, and then we can also see how such a thing can be used, or such a molecule can be used in modern technology. Okay, so that is the idea here. So, okay, so what do you guys know about hemoglobin? Okay, so hemoglobin, if you have taken some biochemistry course, so it is the structure of hemoglobin. So if you take one of these red blood cells, so there are 4 heme units. So they bind oxygen and transport wherever it has to be taken. So they are involved in oxygen transport. Okay, so what is the fundamental molecule there? If you look at that, it's a porphyrin ring. Okay, and if you look at the metal here, so there is ferric ion and ferrous ion. Okay, so that is one of the typical compounds. And if you look at some other biological molecules, such as cytochromes. Cytochromes are also very important. Oftentimes, they're involved in electron transfer. They're involved in catalysis and even in transportation in biosystems or all living systems. And if you replace this iron in these things, okay, then we have something called vitamin B12. Okay, and you know what happens if there is a deficiency of vitamin B12. Right? And if you see in natural system furthermore, even there is nickel containing systems, okay, and they are involved in making methane. Okay, so that means in biological systems, Okay, these molecules are very well established. Okay, so and every time you see this is red now, vitamin B12 also kind of red, and this is some kind of reddish, yellowish red color. Okay, so they are different colors. And if you look at historically what happened to King George III, okay, during his time, he was a British ruler. Okay, during his time, Britain lost, United States or America. Okay, and if you look at, if you read this book, this king was crazy, pretty much. All the time he was angry and mad. So nobody can go and talk to him. Okay, so number one, he's king, so he can punish you to death, right? And most of the time, nobody, So then people realized something is basically wrong with this guy. Okay, then they kind of analyzed, after a couple of centuries of his death, that he had a thing called porphyria. Okay, porphyria. So that is the disease he had. Okay, porphyria has something to do with porphyrin. In biological system, to make this molecule, okay, you need multiple enzymes. It's a multiple step. If one of these enzymes is not functioning the way it is supposed to function, then what happens? You build up the precursors, right? The starting compounds. You build up the precursors, and some of these precursors are not that, helpful. Okay, so every time you build up these precursors, especially something called PBG, that's one of the precursors. So then, you have all the symptoms. So symptoms of porphyria. Okay, so now if you had all those things, you'd also be crazy, right? Just like the king. Okay, so he was crazy because he was having this, disease. Anyhow, so now, not long ago, okay, so all our famous TV actors and actresses, they took care of business. So every time they saw a vampire or werewolf, what they did, they went and, they killed them, right? Now things have changed since then. So now, why kill them when you love them, right? Okay. So the idea is now, so what is the relationship between these guys with the molecule that we are trying to discuss here? Okay. So if you look at werewolves, okay, and this is a deceased person who had this porphyria, okay, and some artist saw that and draw, this is bad, he made it even worse. Okay, these are all fixed. So this is the real guy, and then so somewhere in 1960, a British doctor said, these werewolves are man-made creatures basically. All these people had this disease called porphyria. So they are all kind of disfigured and all those things. Anyhow, this is the reason why, we created so-called the werewolves, or at least the picture. All these things came out of this. Okay? So, what is the truth about these guys? Okay? So, as you know, okay, these vampires are photosensitive. They don't want to see the light. Right? They go crazy. That means they have this crazy behavior. And they have pale skin. And they're anemic. And most importantly, they allow involuntary transfusion of blood. Right? So now, if you look at this, the previous slide, this is just the extension of the previous disease, basically. Okay? But, every time Hollywood has no good ideas, they come up with brilliant ideas. Even a pathetic thing, they can make look great. Okay? So it all started with this famous movie on Dracula. Right? So that is where a couple of decades ago, maybe. Okay? So now we know Draculas are all bad and blood-sucking and things like that. But in reality, But in reality, see, these vampires are four cartopathetic figures, okay, suffering from this disease called porphyria, okay, and we also call them undead and things like that. And with all those things, okay, so if you do research on all those things, so finally their body could not make this heme production properly. They lacked heme, that is your porphyrin. So that's what they loved, all those things. Involuntary transfusion, pale skin, and all disfigured face and whatnot. Finally, that all comes to one simple molecule. Okay? All these things come to one simple molecule called porphyria, or porphyrin. The same thing applies when you have jaundice. Okay, so if a person has a jaundice, so somewhere, the heme breaks down, okay, creating this unbroken porphyrin ring. So now this is not fused, it's broken. And this has that yellow color. Okay, so this is called bilirubin. Okay, so this has that yellow color which is responsible for, jaundice. On a lighter side, here is a cartoon, what our famous guy Simpson would think about this disease. Okay, so here is some parents, and the child has this jaundice, and they think, they may be bad parents, but our guy says, no, they're just fine. Everything looks good. Okay, so anyhow, so that is about heme. Okay, that is about heme. And if you look at the plants now, and go and see what chemical compound which is responsible for this green color in plants, and if you analyze that, there also you find this molecule which is porphyria. Okay, and instead of iron that you had in heme, which was responsible for the red color, so here it's magnesium. Okay, so that makes this compound kind of green, and this also is involved, in photosynthesis. Right? Without photosynthesis is a process where it takes water, carbon dioxide, and make carbohydrates and release oxygen, right? So that we can breathe. And also, if you see what make these fall colors, okay, fall colors you may not see this pretty in Texas, but if you go up north, you will see beautiful colors. Oftentimes, the magnesium porphyrin is because of lack of light, so it is kind of not functioning. It is decayed or degraded. Then what is left in these leaves are other colored pigments, like carotenoids, flavonoids. They have different colored pigments. That's what all these fall beautiful colors we see. Okay? So, if you take a closer look at these chlorophyll molecules, so what we have, so we take in water, light, and make chemical energy, and we produce carbon, or take in carbon dioxide, water, and sunlight, and produce sugar and release oxygen. So this is photosynthesis, basically. So, and if you take a deeper understanding, okay, or deeper look, and see what exactly is happening there, so there are two important molecular fragments there. One is called antenna molecule, which is made out of this green chlorophyll and beta carotenoids. What these rings do, or ringed molecules do, they capture sunlight. Okay? They capture sunlight and funnel that energy to another segment called reaction center, where you have electron transfer. Okay? And electron transfer is very important. Okay? Without electron transfer, nothing happens. Okay? So here we have a bunch of, so light falls, and then we have sequential electron transfer. That means there is electron hopping from molecule to molecule, okay, so that there is no back electron transfer. Okay, so that is the idea. So what happens in a deeper sense? So what we want to do, just to showcase what we want to do what nature does. Okay, this photosynthesis. Okay, so basically what we have is there is electron transfer. That means we have to have two molecules at least. One should be an electron donor, other one should be electron acceptor. Donor means it's rich in electrons, Acceptor means pour in electrons. Okay? Then you shine light on them so that they have electron transfer. And most of the time when you are talking about organic compounds, so they absorb light only a small portion of the electromagnetic spectrum. Okay? So if you want to harvest the sunlight, you want all the light to be captured, right? So that you can utilize the light to the extent it is possible. To do that, what we do, so we have some visible antenna molecules and UV antenna molecules, so that no matter where you shine light, this molecular is not a small molecule, it is a complex molecule. Okay? It ends up in giving an electron transfer. Okay? So here some of the examples that have been developed in my lab for the last 2-3 years. Okay, I'm not going through the details and trying to make you guys take a nap. Okay, that is not why I'm here. So this is just to show there are very complex molecules. So at the end of the day, you shine light, there will be electron transfer. Okay, and this electron transfer becomes very handy. I'll come back to that. And we use different types of material. For example, in the previous lab, we have these fullerenes. They're all carbon buckyballs. Okay, or we can use carbon nanotubes, or we can use graphene, this kind of molecules. Basically, we use carbon-based material, okay, to harvest light energy. Okay, that is the idea. So once you have this, okay, while doing this kind of molecules, so you always come up with pretty good pictures. Okay, so here is one simple molecule. So what we have is like, Star Wars TIE fighter ship. Even, what is this guy's name? Yoda. Yoda is also happy because we can make his shaper, something like that. Okay? Or if you are really creative, so you can come up with a lot of beautiful molecules or structures, and they look good, but also they have a purpose here. It is not just you assemble the molecules and make Yoda happy, but there is a purpose. Purpose being you will end up in seeing an electron transfer. When it does so, these are some of the molecular systems that we have developed. Okay, so when you say they all undergo electron transfer, okay, when it happens that way, ultimately what you want to do is make a device so that you can convert light into electricity. Okay, just like photovoltaic cells. So you guys all know now what photovoltaic cells can do. Okay, now the gas is cheap, so nobody cares, but it goes in cycle. Okay, so in photovoltaic cells, what happens? You have a couple of electrodes there, and you have your molecular system. You shine light. I said it ejects an electron. This electron flows through this circuit. Electron flow in a circuit is nothing but electricity, right? So that is what electricity is. So we make it to flow through this circuit so that, so we use the sunlight and create electrons. Electrons flow through a circuit, then we can close this circuit. So we have a simple solar device, so which can convert light into electricity. And there are a lot of applications. Nowadays people have all these fancy things, especially if you travel in Europe. Every house has this thing. So sometimes they supply electricity to the grid actually. So instead of getting a bill, they get a check at the end of the month. So because they make their own electricity, the electricity is excess, they can sell that, to the grid. Or oftentimes we can also, this is what the cell looks like, your donor acceptor. When you shine light, so you have donor with one less electron, acceptor with an extra electron. So this extra electron moves through the circuit. That's what you create electricity. And then we use a redox mediator to complete the circuit. Okay? So that is how light is converted into electricity. Okay? So, but this all works during daytime. So how to produce electricity? Okay, nighttime. If you have to go only based on renewable energy sources, so you want something, some alternative, when there is no sunlight, right? So we can use the same scheme now the molecular schema I showed, donor acceptors, okay, which will give an electron. On that, what we do, we position a water oxidizing catalyst. Okay, so the role of this catalyst is to take water, okay, and split it. Split it to get oxygen and protons, that is hydrogen plus. Okay, on this side of the molecule, we have something called proton reduction catalyst. There are two catalysts. One is this side, other is other side. And what this does, it captures these protons and it reduces the protons to hydrogen. Okay, now what is that we have accomplished? We have taken this molecular system, which is fairly complex. It takes years to build. Okay, then we take water, split water to oxygen and hydrogen. Now hydrogen is a fuel, right? Hydrogen is a fuel. So we can use this in making something called, something called, what is this? Fuel cell. Okay, fuel cell. So this is fuel cell where hydrogen and oxygen are pumped in, and reverse of water splitting is done here. So hydrogen reacts with water, hydrogen reacts with oxygen to give water. Okay, you have pure water, and that extra energy is what is used to make electricity. Okay, so if you see now all the guys who go to, to moon and all those things, this is the technology they used in Apollo series and all the things. What went wrong with Apollo 13? Have you guys seen a movie called Apollo 13? So what went wrong? They came back, right? The successful, unsuccessful mission or something they call. What went wrong? So one of their fuel cell burst, basically. There are a lot of fuel cells. These fuel cells are used, number one, to make electricity and also generates water. And that is their drinking water. They don't carry cans of water. So they have to make water to drink and survive. So that's what went wrong. So, but anyhow, it is very important. It is very important that, we can make hydrogen using the previous design and convert that into energy here, that hydrogen, by using fuel cell technology. By doing so, we need a good catalyst. So this catalyst should combine this reaction and facilitate that reaction. Okay? So that needs a catalyst. And it so happens that catalyst also is based on this beautiful molecule. And here we have cobalt inside. Okay, this is one of the molecular systems that we developed a while ago. Okay, so we have this nice oxygen is converted into water, right? Oxygen is converted into water. So to do this process, you need 4 electrons. Okay, 4 electrons is needed. But if it is an incomplete reaction, so this oxygen is converted or will be converted to hydrogen peroxide. It is a 2-electron process. Now hydrogen peroxide, you guys have seen that, right? Hydrogen peroxide solution. So why do we use hydrogen peroxide? It's corrosive. It's bleaching. So all the face cream that you use, it has small percentage of peroxide. So that bleaches your skin, so that all the dark spots will be gone and you look pretty. Basically, that is the idea, hydrogen peroxide. But this is so corrosive, if hydrogen peroxide is formed, so there is corrosion of the cell. We don't want that. Okay, hydrogen peroxide. We want water as the product. Okay, so that means you need catalysts which are very efficient, to convert oxygen into water. Okay, so these are some of the things. So then, by using fuel cell technology, so we can make our, transportation easier. Okay, now we have a lot of hybrid electric cars, not in Texas though. So most of the time they try in California. So I don't know why, because they're more susceptible, I think. Okay, so now this compound that Fortune I was talking is also a beautiful drug. Okay, so in this drug, so basically there is a therapy called photodynamic therapy. This is not like radiation therapy, okay, or chemotherapy. Okay, radiation therapy uses gamma rays. They are very powerful. Okay, they kill good cells. They also kill bad cells. Okay, but here what happens? We have this compound. They are very colored. Colored means so you can use visible light. Say for example, I'm using here red laser light. Okay, so whenever you hit this, so now we have this tumor. Then we inject this compound which accumulates on the tumor. Then we shine light, and this light, in the presence of oxygen, okay, it kills this tumor. Okay, and the same idea is used in photorejuvenation. So I have a person with a lot of acne scars, and we can inject such a drug here, okay, and make this person sit in front of light, and at the end, you have a pretty good looking face now. So that is simply because this simple compound. Okay, so it is like it's a drug basically, right? It's a drug, so which helps to cure all these acne marks and such. Okay, and we develop these things. We develop these things in our lab because I have a very good synthetic lab. Okay, and I'm a chemist, so I don't do studies. These studies will be done. These are some of the drugs that we have developed for this photodynamic therapy application, which are being investigated at Roswell Park Cancer Institute. That's in New York, Buffalo. Okay, and he's the guy who's developing or studying, and he's so famous wherever he goes, so a band follows. Okay, so anyhow, so let's talk about the sensors and chemosensors, biosensors and such. Okay, now the sensors field, basically it is about $5 or $6 billion market worldwide. Okay, so you need a sensor for pretty much all the applications. And finally, you need a sensor for a sensor itself. That means there is such a great demand. Okay? So one of the ways we develop, because these are all colored compounds, okay? So for example, this is one of the molecules, still based on phosphine ring, but it has different substituents. And these hydrogens here, they bind to anions. Okay, anion recognition is very important, especially nitrates, phosphates in soil samples, right? So they are very important. So we have color indicators, okay, which are very specific to some of these anions. This is one example I'm showing. This compound, in presence of so many different anions, they are different color. We can pretty much have the entire rainbow colors. So that means if I suspect iodide, so in propionic acid, if it is yellow, okay, everything else is red or pink or magenta, so I know there is iodide in solution, something like that. Okay, and if you want little more sophisticated, that is not very specific, right? If you want something little more specific, then we have to engineer the molecules. Okay, it goes this way. One of the things that we have developed is called imprinted polymers. Okay, imprinted means you make a three-dimensional, like I was just told that three-dimensional printers. You guys made a world record here. Okay, so here is also something like that here. Okay, but your printer is very big, but our molecules are tiny. Okay, but they work. How it works? Now I have a template molecule here. Okay, if you are after analyzing a compound, this template molecule is itself your analyte. And this molecule on its periphery will have some functional groups like carboxylic acid or hydroxide, amine groups, something like that. Okay? Then what we do, we will create some functional monomers which will complementary bind to these active sites or functional groups. Then we will have something called a cross-linking polymer, okay? And then we polymerize the entire thing, either photochemically or electrochemically, okay? And then by some mechanism, we get rid of your analyte. Now, what is that you have created? You have created a three-dimensional pocket, right? Which has binding sites, which is meant for this molecule. right? It is so specific. So that is the technology. So we have developed this for many applications. One of the applications in the beginning we studied was nicotine. Okay, so nicotine, all the tobacco problems you guys know. It has every possible problem you can imagine. Okay, and still, all said and done, those who, there are some odd people who live up to 100 years even though they are smokers. Okay, there are always, we say that, all the problems, but still, people live longer. I'm not trying to promote smoking though. Okay, so one of our thing was to make a sensor for this, so that if you have a smoker, okay, nicotine you can find in their saliva or all the things, so we can detect, or in the air sample in a closed atmosphere like a plane carrying people, if somebody is smoking in the toilet there, so we can detect that. So idea is this molecular imprinting. So here is the compound that we use. Here also we have a porphyrin. Okay, this is also beautifully colored. And then we have some functional groups which could be electropolymerized. Okay, and that once you polymerize with this template, this is your nicotine, it binds very well. And then what we do, so we generally weigh the sample. Okay, we can detect in many ways. One of the simple ways is weighing them. So how do we weigh these samples? It is a quartz crystal, basically. Quartz crystal. Quartz crystal is what you have in your wristwatch. Okay, these crystals, they vibrate, and every so many vibrations is equated to 1 second. Okay, if it is 10 megahertz, so it's 1 second, pretty much. Okay, and every time you have some load on the crystal, the vibrations slow down. Okay, so that means we can equate the change in frequency to mass on this crystal. So that is quartz crystal nano balance. How accurate it is, we can get nanograms. Nano being 10 to the power of minus 9th of a gram. Okay, so that is the technology. But there are different ways, but this is one of the simple ways that my undergraduates do in the lab. Okay, something like that. So every time you have nicotine, there is a change in frequency. Okay, and that is equated to mass change, basically. That's what I'm trying to show. So this is one way of making sensors for, different analytes. They're all based on our primary molecule, which is porphyrin. Okay, so for what I have shown this molecule, porphyrin, which is a very important component in biology, right? In biology, like photosynthesis, heme, cytochrome, vitamin B12. And it is also important as materials, okay, to design new materials for solar energy harvesting, photocatalysis, optoelectronics, molecular switches, logic gates. I did not give a lot of examples on the later part because simply there is no time. And also this is a drug also. Okay, it's a pharmaceutical drug where this is a modern thing now, photodynamic therapy, where you can use visible light instead of gamma rays. Okay, and finally this is also used in technology. So where you can make sensors, biosensors and such. Okay, so the rest of the talk is just to promote undergraduate research for the last 10 minutes or so. Okay, so what is research, by the way? Okay, so now this is only for those who have interest in research. It doesn't mean that you can go out. Okay, I'll be done in about 5 minutes. Okay, so what is chemical research? So you all know, discover new compounds and the properties. Like I showed one example based on porphyrin, how many directions we can go, right? Discover how the new compounds function. Publish findings worldwide, so that others doing similar work can learn from it, question it, collaborate, things like that. And research sometime leads to recognition. Maybe we will get a Nobel Prize in chemistry. If not, at least you are promoting advancement and other perks. Okay? And if not, whatever you publish It adds to the collective body of knowledge, okay, which you may not see the use right now. So later somebody will find use for that. So it's important you do research and do not lock it up. You publish, make that knowledgeable, available for everybody. Okay, so what it takes to be a research chemist. Okay, so first you have to graduate from high school, which you all have done that, right? Then get a bachelor's. Now you guys are in the process of getting there. Okay, so then you will choose one of these majors: chemistry, physics, chemical engineering, biology, computer science, electrical engineering, a lot of things. Apply to graduate school. They usually will pay some money. So it is not a burden on your parents. Okay, but that is just enough to survive. Not a whole lot. Okay, so pick a program and do teaching, research, and other activities, defend a thesis, then you become doctor. Okay, so it is that simple. But it takes about 5-6 years. Okay, what are the benefits? If you did choose to do that, so what are the benefits? So in your lifetime, you will make more money than anybody compared to those who have bachelor's or master's. Okay, you'll most likely be a specialist or a manager or some important position. Okay, so you get to travel. You need not be in this small town all the time. So, and you will live to an old age. That is true. Especially chemists, their lifespan is a little longer than non-chemists. Okay, so, and your job is not a dead-end job. So this job is interesting, and it's always changing. That's what makes it interesting. It is not the same thing you do every day. Okay, so this is my last slide. Okay, so why UNT? I come from UNT. Okay, so this is a big school. We have a lot of students, 36,000. We have about 1,200 faculty. We have 97 bachelor's program, 82 master's program, 35 doctoral program. You can pick and choose whatever you want. And chemistry, we have top-notch faculty, cutting-edge research, okay, and a lot of sophisticated instrumentation, things like that. And also, if you look at jobs, Dallas area has a lot of jobs. Okay, in fact, we are yet to graduate an unemployed graduate student. So that most of our students have something before they graduate. With that, I'd like to thank you guys for being here.
I think we have time for a few questions from the audience.
So do people who have porphyria, did I understand that what you said is they do actually suck blood?
No, they don't suck blood. Sucking blood is a man-made thing. Okay, so those who have porphyria, they have deficiency. Okay, and that deficiency, if is not treated properly, okay, then you have all these anemic features. Okay, they don't drink blood. Okay.
So they just look like.
No. Don't do that.
No, I won't.
Okay. Other questions? Yes. Right.
So, you were saying that with the proper chemistry, a hydrogen fuel cell is a possibility that is more feasible than the former electrolysis used to create hydrogen.
Water splitting is done basically by electrolysis to begin with. But here it's a reverse layer, where hydrogen and oxygen are combined to give water.
Okay.
That is the fuel cell. And when it's so exothermic, the reaction is very exothermic, burning hydrogen. Okay, that energy is what we converted into electricity. Okay? It's exactly opposite of electrolysis. Yes, please.
Have you tried the scorpion and other drug except then
No, the photodynamic therapy is a popular cancer therapy right now. And there are at least 20 or 30 groups all over the world, including ours. And we change the properties so that it can selectively bind to tumor, not the healthy cell. And there is something called single oxygen production, because the oxygen we breathe in is a triplet oxygen, that is harmless. But in the presence of light and this compound, this oxygen is getting converted to triplet oxygen, singlet oxygen. And this singlet oxygen is the killer of the tumor. So that means any drug we make, the first thing we do is whether it can produce singlet oxygen. Okay? So there are a lot of designs, there are a lot of principles, there is a lot of theoretical work, and there are a lot of advanced studies also on this thing. So every permutation and combination is being tried out, because at the end of the day, there are 2 or 3 drugs already in the market. Okay, but to bring out one drug, it costs about $300 to $400 million by the time you get FDA approved. It's not you make a compound and you have it. It has to undergo so many tests, but it's a combinatorial approach. That means we do many things and then do studies and pick the best one, and from there we do all systematic studies.
Okay, if you have more questions, our speaker will be down here for a few minutes, and also, feel free to join us for lunch. Okay, let's take the class.
So how do you kill this?
Oh no, don't kill it yet, because I gotta stop this first.